Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Marianne Jelinek, Associate Director of R&D at Active Motif, on this show. Usually I introduce the guests on this podcast, but this time I want to pass the question to you. Can you give us a short introduction of yourself?
Thank you. Certainly. As you mentioned, I am Associate Director of R&D here at Active Motif. My background started with a PhD from McMaster University after doing an undergraduate at McGill University. I've received a postdoctorate degree, training at McMaster University in cell signaling, and then went on to work in biotech. I have been working in the field of epigenetics since the early days, back when the first histonoacetyltransferase was discovered, and had the opportunity to be involved in the commercialization of the first chip kit.
Yeah, maybe we can talk about this at a later point when I come to my next question, because before you went to university and before you started your career in biotech, what was it that made you interested in biology in the first place?
In high school, I was always interested in the sciences, and I found myself leaning towards the biological sciences. I also was fortunate enough to have 3 summer internships, both before starting undergraduate and throughout the 3 years undergraduate program at McGill University. 2 of them were involved in DNA repair labs. One in E. coli, one in fungi, and then I went on to a biochemistry lab, again in looking at proteases involved in regulating DNA repair. And that was enough to spark my interest that I really loved the lab setting and the biological sciences. And most of my career has been involved in either cell signaling from receptor tyrosine kinases or transcription.
Mm-hmm. So usually we talk to people that are still in academia, and you are one of the few ones that are not in academia anymore. So what influenced your decision to move away from academia?
A job offer.
So money.
Money. Well, no. So after completing my postdoctoral training, we were already starting a young family, and we wanted to have a work-life balance. And it was just convenient to be able to have that, sort of have the best of both worlds, have a family, have a career in science without having the stress of being the sole funder of your own career through obtaining grants. And that's fundamentally what happens in academia. You might be brought in as an associate professor, and there might be some starter funds, but then you're expected to fund your own career through grants. And it turns out that biotech is not all that dissimilar. A lot of companies will have their standard portfolio of products and the product types that they want to develop. But then new ideas come in that are a little more risky and may require longer time to develop. And that's where grant writing becomes of value even in biotech as well. And that is how I got into grant writing.
Maybe you can elaborate
on the biotech side.
Yeah, maybe you can elaborate on this a little bit further, because I always thought that grant writing is only for people at universities, and now you just said that grant writing is also a part of your job in the industry. So how does grant writing fit into your job in the industry?
So as I mentioned, when there is a project that is deemed by the company to be a little bit too risky, would require too much resources, and it's sort of deemed as, well, we would only do this if you could find external resources. And it turns out when you have a look at the funding agencies, they all have, or quite a few of them, especially the NIH, will have specific categories of grant classes that are specific for industry. And these can be small companies. And then the bigger pharmaceutical companies, the big drug development companies, they also have an opportunity to apply for grants. So these grants are not restricted to academics. And so you have grant opportunities that are both run by the governments of countries. So here in the United States, we have the NIH, but there are also other organizations that offer grants. So for example, NATO has a granting mechanism as well, where they fund research that's of interest to the security and the safety of NATO member countries.
Would this then mean that if something comes out of it, you need to be in NATO part of it? Or the organization that gives you the grant, for that matter? Yeah.
So I'm not familiar too much with the NATO funding, what the restrictions are. Typically, I can speak certainly for the NIH. The companies that develop the grant own their own technology. They own their own intellectual property. It is only under extreme circumstances when, if there is a need, some sort of state of emergency declaration of that nature, that the NIH could then come in and say, oh, we need this technology for a particular purpose. And they might broker some sort of deal with the company that developed that platform. But for the most part, the technology is the property of the company that's developed it.
Mm-hmm. Have you found that there is a difference between the grants that are awarded to academia and those to companies? Is there different, for example, in money or in the regulations that you need to fulfill?
Yes, there definitely are differences. The typical business is strongly discouraged to apply to the academic funding. That's the grants that are geared towards the academicians. And the reason is academic institutions tend to have a much greater depth of resources and facilities, that core laboratories that a small company would not. And specifically here in the US, by separating out a separate budget, a separate allocation of funds for small businesses, the intent behind that is to foster and promote growth and innovation within the United States, to take academic findings that have commercial value and encourage academicians to start their own companies and see what they can do with their finding, their product, their idea, and turn it into a business. And this sense of entrepreneurship is there, and these grants are specifically set aside for companies as to protect them, to make funding of the ideas a little bit easier than would otherwise be happening in the academic setting.
So when you started writing grants, what type of grants have you done?
So as a graduate, as a graduate student, there were fellowships, and there was a lot of mentorship that went on between myself and the PI, and those fellowships were then, to help, pay your living expenses. In the biotech sphere, I've had experience writing what are called the Small Business Innovative Research Grants. Those are the SBIRs, and they tend to be three phases. There's the proof of concept phase, and that's the phase one, that tend to be shorter in duration, 6 to 12 months, smaller budget. Then there's the phase two, which are the actual R&D, prototype development, validation. And then there's the phase three, which is commercialization. The NIH specifically tends to focus on phase one and phase two. We've also written grants and that have been funded by the National Science Foundation. So that's a different agency here in the US that is more heavily on the engineering side as opposed to the NIH, which is more focused on human health.
In academia, it's very, I guess, straightforward to come up with ideas because it's very project specific, right? So you want to get the money for your idea or your project or your PhD or postdoc, whatever you're doing. But how is it in the industry? How do you get your ideas? How do you know if it's a one that will support or is solid enough?
So there's no difficulty in finding ideas. As researchers, we attend meetings, we hear academicians speak, we have our own ideas from research projects that are going on. So in bigger companies, there's an actual research lab that is not too dissimilar from an academic lab. Smaller companies, it tends to be more development oriented, but you get your ideas, and then you can have a look to see which institute within the funding agency is interested in that field of research. So within the NIH, there are several dozen institutes, and they publish on their website lists of research areas, research topics of interest to them. And so you can peruse this document. In the case of the NIH, it's several hundred pages long, and you can identify where your idea fits. The next best thing is to actually contact the scientific leads at the institutes just to find out whether or not your idea is a good fit for their particular funding institute. Research institute, and they'll give you candid feedback. You provide for them a one-page summary of the background and the main goals of what your research plan is, and they'll give you feedback, and they'll say, well, that sounds like that's a better fit for that particular institute, or they'll tell you that it's a good fit for them, and they'll often give you feedback and guidance to strengthen your proposal verbally, because they want to see funding applications be successful.
Mm-hmm.
And, so there's this whole infrastructure of these grants management people who are there to take the taxpayer funds that have been allocated to them and distribute them in the best manner possible. And from their perspective, the best manner is funding projects that have a high likelihood of impact, positive impact for human health in the case of the NIH, and also success of coming to market. So they're there to help you.
Mm-hmm.
The program officials are there to provide guidance to the grantee as to what they're looking for, and provide pointers if there's any deficiencies in the approach.
So how would you then handle this feedback? Would you go back to like your company, your bench, or rather the drawing board, and maybe also work on the idea itself?
Oh, definitely. The best thing to do is to speak to your teammates, your collaborators, to incorporate the feedback that you've received and to strengthen your idea. There's several areas of focus that make a project fundable. It has to be significant. It has to basically have some innovation. There's got to be something new to this. You're never going to get funded for trying to do something that's already well established. scientifically. And so there's two fundamental questions that have to be addressed when writing a grant. And you have to convince the reviewers that A, the project is doable, that it should be done, and 2, that it can be done. And if you can convince reviewers that both of these are achievable, then the likelihood of being funded is very high.
Maybe we can take one step back for a little bit. So, because now we were already talking about reviewer feedback, but how long does it take before you feel confident in your grant writing and submitting your grant? Like the idea has been in your head or has been discussed among the team, and now you want to submit a grant. So how long can this take?
The length of time from going from idea to submission can be as quick as 4 weeks. The confidence in whether or not, the likelihood of being funded, there's always the case of the nerves. Did I cover all the possible arguments in favor to justify the rationale that this is necessary? Did I forget something in my approach? Did I leave something unaddressed that could be easily viewed as a criticism or a weakness? So the length of time for writing a grant can be pretty quick. The more people that you involve, the better on a grant. So in a business, you typically have a specific area of focus of expertise. And as soon as you go outside of that area of expertise, you want to show that you have collaborators that can support you through their strengths. And so oftentimes having consultants, academicians or collaborators from other companies is viewed as a strength when you're proposing to do a project that involves multiple disciplines and a particular company might not be viewed as an expert in that field.
So you have your idea, right? And what is it that you need to prepare usually in this grant writing process to go from idea to submission?
All right. Well, a lot of that depends on the funding agency. They will have a list of documents that are required. Oftentimes, so you have your research plan, and the research plan will include the significance. Why is this important? And then the innovation, what's new? And then there's the approach. How are you going to go about completing this research project? And depending on the grant, if it's a phase 1, that research plan with the significance, innovation, and approach is 5 pages long. If it's a phase 2, it's 10 pages long. So it gives you a lot more space to develop your approach. There is often ancillary documents that, in the case of an SBIR, the equipment that is at your facility, the additional resources that you have with regards to IT infrastructure, freezers, fridges. There's also a biosketch, sort of kind of a resume, but more scientifically slanted with all of your publications, your research interests, your contributions to science. There will also be an abstract that is published upon and available in the public domain once a grant is funded. And then there are budgetary documents and some regulatory documents that, depending on the nature of the agency, will have specific lists of questions that need to be answered. So, for example, for the Small Business Research Initiative grants, they want to make sure that you really are a small business that qualifies. And so you have to answer specific questions and be listed within the Small Business Administration that here in the United States.
And they probably also want to have like a time frame when you're going to be finished with that, right?
Right. That is part of the research plan, sort of a timeline and if there's any interdependencies between the various milestones or goals of the project. A phase 2 grant, because that's more advanced with regards to its product development in the life cycle of the project, that also requires a commercialization plan that's up to 10 pages long, and that's more market focused. Who are your competitors? How are you going to protect your intellectual property? What else is already out there? What kind of revenue do you expect to have from the product? And so that then involves not just the R&D people anymore, that now involves sales and marketing to help develop the commercialization plan for those phase 2s. Mm-hmm.
So in academia, you also have to be a reviewer for other grants, and you basically get no money for that. Have you ever served as a reviewer for other grants?
Yes, being a grant reviewer is not something that is restricted to academicians. As soon as you have received a grant and have been funded a grant, you're a PI on a grant, you're expected to pay it forward by being a reviewer on other for other grants. And it's actually an extremely valuable experience because it teaches you, helps to refine your grant writing skills, because now you understand what goes on after you've submitted your grant.
So how often does this occur that you are contacted to be a reviewer? Is this like if you've done one because you have received one grant, then you are done, or are you contacted more often than that?
You can be contacted up to 3 times a year. So in the case of SBIRs, there are 3 funding deadlines, and depending on the volume of applications that are received and the nature of applications, when you submit your biosketch, this becomes available to the funding agencies, and they are able to see where your area of expertise lies. And so you will be contacted by different program officials to say, hey, we are looking for somebody who has experience in epigenetics. And we have 6 to 8 grants for you to review. Mm-hmm. And, if you are available, so there's been a big change before COVID and after COVID. Now there's a lot more of these grant reviews are done remote. Prior, the people would have to fly to the NIH, which is in Washington, D.C. So here on the West Coast, you would end up losing a full day's travel on each end. So now that they're comfortable with remote meetings, and we do basically Zoom meetings, and you're given a few weeks to read through the grants. There's a form that you fill out where you indicate the strengths. And here in the NIH, there's 5 categories. There's the significance, the investigators, the approach, the environment, and innovation. Those are all factors that we have to indicate the strengths and the weaknesses. And then we also put together an overall summary of the overall impact. And that impact statement is really the culmination of those 2 questions that I mentioned earlier. Can it be done, and should it be done? And if you can argue that favorably, strongly for both of those, that really helps to drive the score up towards where it's fundable.
I see.
So after,
go ahead.
So at the actual study section, which is the meeting where 50 or 60 grants are reviewed, each grant is typically reviewed by 3 individuals, and there's 15 to 20 minutes allotted per grant. What's happened since COVID, when a lot of people had downtime and spent a lot more time writing new grants, there's been a higher number of grants to review in one of these review sessions. And so it used to be that all of the grants could be reviewed in one day, and it now has trickled over to where it's required 2 days. Maybe not 2 full days, but you do 4 grants in an hour. So when you have 60 or 80 grants, you can easily see how that spills over to 2 days.
Yeah, definitely.
Yeah.
So
Yes.
since you have gone through this process many times, probably, or sometimes, what other tips do you have to consider in this process?
With regards to as a reviewer, or how
As a writer, yeah.
As a writer, so clarity of what you write is very important. Being concise and clear. Reviewers have general backgrounds that might not exactly overlap with what you are proposing. And so you have to be very careful to define terms and explain things at a point where somebody who might not be an expert in your particular field can still understand the significance and how and the approach. It's really impressive to be a reviewer on a grant of somebody who's had a highly successful career, and to see how well written those grants are. Very straightforward, short sentences, no ambiguity, no opportunity for misunderstanding or misinterpreting what the intent is, and very clear. And you walk away after reading that, like, I know precisely what that person wants to do. They are able to paint a picture for you of precisely, experiment by experiment, what needs to be done, and how one experiment will flow to the next. And so if you ever have an opportunity to review somebody's grant as sort of to get as if they ask you for extra feedback, or if you know somebody who is like a leader in the field, and if they're willing to let you see one of their grants as an opportunity to sort of see what a well-written grant looks like, by all means, jump on that opportunity, because the style of writing is very different from grants. You have to be sure that you're concise and clear, and you're given time, page limits in which you have to fit your ideas. One of the biggest tips that I received when working with an academic collaborator is, if it's complicated, turn it into a diagram, turn it into a figure, and let the figures lead the reviewers through your process, because it's much easier for people to see and understand through a graphic than it oftentimes is to explain it into words. And I think that was the best advice that I've ever received. Was there's that old adage of a picture is worth 1,000 words, and in the case of a grant, definitely so.
So you also mentioned that you have to protect your IP, right? And patents are very important in companies. So how does that fit into grant writing?
It is expected in most of the grants that a lot of the information that's in there is confidential. There are opportunities for the grantee to flag specific sections that are deemed to be proprietary, and the entire grant review process is highly regulated with regards to confidentiality. Reviewers are agree to a code of conduct of confidentiality, and not sharing any information, not even discussing any grants with any of their colleagues, any other reviewer. It's strictly trust that's built up. And if there are any indications of anybody trying to breach that trust, we've been given specific instructions on how to deal with that. So, for example, if another reviewer contacts you and say, "Hey, I know that PI, could you give them a favorable reference?" We immediately have to disclose that inappropriate contact to the agency that carries out the review to report it. When, as reviewers, we are first given the list of grants that are going to be under review, the first thing we have to do is to declare conflicts of interest. And the conflicts of interest can be declared both at a company-to-company level or just PI to PI level. And at any point, and as soon as you declare a conflict of interest, that individual is not part of that review discussion. They get put into the timeout room, if you will, where you lose access to the discussion and access to the review. So that helps to preserve that confidentiality, so that if there are, competitors or any conflict of that nature, that there is that protection of proprietary information. Even if at the start of the discussion of a grant, at any point, if you realize that you have a potential conflict of interest, then you alert the officials managing the review session, and they'll immediately put you out of the discussion to protect the integrity of the review process.
Interesting. So I've reached the end of my questions. Did we miss something important, or would you like to add something?
So grant writing, I think most people have a love-hate relationship with the whole experience. It's challenging. It can be frustrating, but when you are successful and get funded, the frustration is all sort of behind you. And the frustration is not so much in the grant writing itself. It's you have a lot of forms and fields that you have to complete, and sometimes there's a specific format that the agency requires you to use. So one example of that is, to avoid confusion, we have this 5-digit postal code or zip code, and at one point they switched from the 5-digit code to a 9-digit format. And if you didn't have the right 9-digit format in there, that would delay the submission of your grant. So that's where the frustration comes in, is when there's these bureaucratic issues that pop up that prevent you from getting your grant submitted in a timely fashion, that it becomes very, very stressful. Yeah. The best thing to do, the best piece of advice to do that I have is share your ideas with your colleagues for your grant proposal. If they can find areas that to strengthen the clarity, the ease of reading, take those suggestions to heart, because as I mentioned earlier, the reviewers are not going to necessarily be experts in the specifics of what you're proposing. So the more clear that you can write the grant, the better. And then also to always submit the grant 2 to 3 days before the deadline, so that if there are some glitches in the computer systems that you're submitting through, that all of your work does not get wasted, and then you're delayed, and then you have to wait for the next granting cycle.
Yeah, I can remember the academics that worked until the last minute of the deadline and then submitted it, and most of the time it got through, but sometimes it's really frustrating when it doesn't.
Right. And also the quality of the grant tends to be lower when it's a last minute scramble, because it becomes apparent when certain documents are rushed and are not as clear, as opposed to a grant that's submitted 2 or 3 or even a week in advance. Best thing to do is to sort of start from the deadline, work backwards, and sort of make yourself a checklist of what needs to be done and by when, and get feedback from your colleagues and also the funding officials as to how, to make sure that what you're working is in line with what their interests are.
Yeah.
Because as I said, they're interested in handing this money out. That's their goal, is they have this money, they need to spend it. And, because if in the next year, if in the budget they're asking for more funds, but if they haven't spent all of last year's funds, they're not going to be able to ask for more funds.
Yeah.
So, that money is there for us to use to develop innovative products. And it just behooves us to write good proposals to convince others that our ideas are sound and would advance scientific knowledge and discovery.
Yeah. Speaking of that, as you said in the intro, and maybe we can circle back to the intro, you were involved in developing the first chip kit back in the days. Could you talk about this and maybe how kit development evolved over the years?
Sure. So in the early days of epigenetics, a lot of work was being done in yeast, and we had an opportunity to, again, small companies collaborating with academicians is the best path forward. The PI who had developed chromatin immunoprecipitation in yeast wanted access to some really good, well-validated antibodies. And so it was just one of these swaps. Here's the protocol. We'll give you some antibodies. And in the early days, when people were looking at just locus-specific mapping of transcription factors or histone binding sites, the readout was qPCR. But as the years have advanced and technologies and next-gen sequencing platforms have come in, these chip kits have had to adapt in their protocol and their methodologies, even in something as simple as switching from agarose beads to magnetic beads to speed up and facilitate the sample handling process. And so it's important for companies that develop assays and technologies to keep up to date with the advancements to make sure that the overall readout is compatible with the technologies that are now available.
Yeah, that's true. Everybody's doing ChIP-seq now.
Correct.
Yeah.
Correct.
So
and it's no longer just ChIP-seq. There are easier, faster alternatives to ChIP-seq. There's now cut and tag and cut and run, and people are exploring those because if you can get an experiment done in half the time, then it gives you more time to do something else. And that's why Active Motif continues to develop different iterations of these products, because there are technological advances that we would like to incorporate to help people get science done easier and faster.
Yeah, I guess this is a good point to end this interview. Thank you, Marianne, for your time and for being on the show.
My pleasure. Thank you, Stefan.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.